Assalamu alaikum. My name is Nimra Razak from MS Chemistry Batch 2. My student ID is 2. Today I am going to present my presentation topic, which is microbial conversion of coal, to Dr. Hammad Majeed, Head of Department of Chemistry, University of Management and Technology, Sialkot. Here is a look of the table of contents. The table of contents includes overview of coal, major coal deposits in United States, Western region and Eastern region, microbial conversion of coal, then comes abstract of the article, introduction, microbes used for conversion, measurement of the biogas produced, materials and methods, utilization of coal washery rejects by different microbial sources for biogenic methane production, then comes improved formation of biogenic methane by cultivable bacteria in highly volatile bituminous coals. Then comes conclusion, last, future trends, and then comes references. Firstly, we will see that what is microbial energy conversion. Microbial energy conversion is basically the shorthand term for technologies like these. In microbial energy technologies, microorganisms make fuels out of raw organic materials, thereby converting the chemical energy in the biomass into chemical energy in the form of ethanol or hydrogen. What is microbial communities in subsurface of coal? Seams are responsible for the conversion of coal organic matter to methane. This process has important implications for both energy production and our understanding of global carbon cycling. This all comes under the overview of microbial conversion of coal. Let's discuss in detail. Firstly, we will discuss abstract. High energy consumption and depletion of fossil fuels results to the introduction of new technologies to produce alternative fuels with fewer emissions of greenhouse gases. The present investigation was focused to utilize the waste coal washery rejects as a substrate to produce biogenic methane under optimum conditions. Coal bearing sediments are major reservoirs of organic matter potentially available for methanogenic subsurface microbial communities. Experiments were performed to explore the efficiency of non-coal samples and coal mines enriched samples in the degradation of coal washery rejects. Further, cow dung, anaerobic sludge, and coal washery rejects were taken at various concentrations to develop anaerobic slurry and analyzed for its biogas production. The anaerobic slurry, which contains 1:1:1 of cow dung, distillery anaerobic sludge, and coal washery rejects, produced methane of around 55.7%. The coal enriched samples show a maximum of 22.6% of methane. Subsequently, the best methane producing anaerobic non coal consortiums were compared with coal enriched microbial culture in converting coal washery rejects of 10 gram per liter to methane. Results revealed that cow dung inoculum and coal mine enriched inoculum source produce the nearly same amount of methane. The selected anaerobic slurries and coal-enriched samples can utilize subbituminous coal washery rejects in methane production. Thus, these consortiums can be applied in converting a large amount of coal washery rejects into methane, thus can lead to the reclamation of the site. Then comes introduction of the article. The rapid increase in the consumption of energy resources leads to the exhaustion of conventional fossil fuels. It was generally believed that coal sources are not favorable as living habitats for microorganisms due to their recalcitrant chemical nature and negligible decomposition. However, accumulating evidence has revealed the presence of diverse microbial groups in coal environments and their significant metabolic role in coal biogeochemical dynamics and ecosystem functioning. Coal is extremely rich in complex organic matter and therefore a very attractive carbon source for microbial biodegradation and eventually for methanogenesis. Lignite coal is soft brown sediment with high water content and a relatively low heating value. Under the development trend of global green energy and low carbon transition, maintaining energy security and coping with global climate change have become major challenges for the world. Considering the cleaner combustion characteristics and higher calorific value of coal bed methane, it has gained much attraction compared to coal. The production of biomethane from coal includes three processes. The first process is release of soluble intermediates from coal, then degradation of the soluble intermediates into the substrates that can be used by methanogens, and third, generation of methane. The presence of effective microorganisms that convert available carbon in coal to methane and environment support the reproduction and metabolism of the microorganisms. Converting biomass organic matter into biogas is an inexpensive, highly feasible, and environmentally friendly treatment method. Biomass with carbon neutral properties can reduce greenhouse gas emissions during energy production. The co-conversion of coal with straw can increase methane production several times compared to the bioconversion of single coal or straw. Geochemical investigations were combined with microbiological and molecular biological approaches, leading to the identification and characterization of the bacterial and archaeal community composition in coal rich sediments and groundwater samples, as well as methanogenic enrichment cultures with hydrocarbons as sole carbon source. Integrating these methods, we were able to show a close interaction between the organic substrates and microbial populations in coal-rich sediments with the groundwater aquifer system. Which microbes can be used for the conversion? Microbial communities in subsurface coal seams are responsible for the conversion of coal organic matter to methane. This process has important implications for both energy production and our understanding of global carbon cycling. Despite the environmental and economic importance of this process, little is known about which components of the heterogeneous coal organic matter are biodegradable under methanogenic conditions. Similarly, little is known about which taxa in coal seams carry out the initial stages of coal organics degradation. To identify the biodegradable components of coal and the microorganisms responsible for their breakdown, a subbituminous coal was fractionated into a number of chemical compound classes which were used as the sole carbon source for growth by a coal seam microbial community. The 65 microbial taxa has identified that were able to proliferate on specific coal fractions and demonstrates a surprising level of substrate specificity among members of this coal degrading microbial consortia. Additionally, coal kerogen, the solvent insoluble organic component of coal, often considered recalcitrant to microbial degradation, appear to be readily converted to methane by microbial degradation. These findings challenge our understanding of coal organic matter catabolism and provide insights into the catabolic roles of individual coal seam bacteria. The materials and methods that were used in the article are: firstly, we will discuss the method 1. Method 1 includes the utilization of coal washery rejects by different microbial sources for biogenic methane production. Feedstock coal washery rejects or the coal washery reject sample was obtained during washing the subbituminous coal to reduce the ash percentage present in the coal. The sample was dried and ground using mortar and pestle. The coal which passed through 60 mesh sieve was used as a substrate. To compare the geochemical properties of coal washery rejects with the raw coal available in the mining area, the raw coal was collected from the site. Collection of inoculum source. The inoculum sources collected for the study was cow dung and distillery anaerobic digester sludge. Fresh cow dung was collected to use as the inoculum source. Generation of enrichment cultures. Two different experiments were established to generate the methanogenic consortium to use the coal washer rejects as substrate. Several non-coal microbial sources have the potential microbial consortium to produce biogas from complex polymers. So, in one experiment, the most recognized biogas producing inoculum sources, cow dung and distillery anaerobic digester sludge, were used along with the coal washery reject. This was performed to create the non coal consortium with the capacity to degrade coal washery rejects. The biogenic methane in coal beds is produced by the microbial communities present in the coal bed seams. The coal bed consists of the potential microbes to convert coal to methane. The other experiment focuses on the isolation of the microbial consortium for methane production from the coal samples obtained from the coal mines. The coal washery rejects and mixed cultures were designated as CR3, CR4, CR5, CR6, CR7, CR8, CR9, and CR10. The modified mineral medium was prepared which contains the following minerals: KH2PO4 0.25 gram per liter, NaCl 0.5 gram per liter, MgCl2.6H2O 0.2 gram per liter, CaCl2.2H2O 0.025 gram per liter, NaHCO3 0.2 gram per liter, and yeast extract 0.5 grams per liter. For enriching the growth of methanogens from the coal, sodium acetate, methanol, and hydrogen ratio carbon dioxide were added as a substrate which stimulates the production of biomethane. Acetoclastic methanogens converts sodium acetate to methane. Similarly, methylotrophic and hydrogenotrophic methanogens convert methanol into methane. These are the direct substrate of the methanogens. So, the addition of these direct substrates will enrich the activity of the methanogens, which will further easily utilize these intermediate compounds produced from the coal during the process of anaerobic digestion. Here are some reactions that involve the methane production. Carbon dioxide plus 4H2 reaction turns into methane plus water. CH3COOH reaction turns into CH4 plus CO2. 4CH3OH reaction turns into 3 methane plus carbon dioxide plus water. This confirmed that the selective activation of the coal seam bacteria played an important role in the enhancement of the biogenic coal bed methane, especially that of cultivable bacteria, which has important value in the coal bed methane utilization. Methanogenesis by the methanogenic archaea is currently the refresh focus of most of the studies regarding biogenic coal bed methane. Firstly, all the experimental bottles were pressurized with a nitrogen ratio carbon dioxide is 80 ratio 2 gas mixture to create a complete anaerobic condition. All the samples were incubated for 40 days at 37 degrees Celsius and shaken every day for 2 minutes. The percentage of methane and carbon dioxide in the headspace gas produced was quantified. The pH after anaerobic digestion was monitored. The cultures which produced maximum methane were selected for further experiments. The potential of the selected cultures was analyzed by adding them as inoculums for coal washery rejects. Then comes the measurement of the biogas produced. The composition of methane in percentage and carbon dioxide in the gas produced was analyzed using a biogas analyzer. The methane and carbon dioxide were analyzed by a dual wavelength infrared sensor with the reference channel. The analyzer was calibrated using standard gas mixture, where mixture 1 contains 5% methane and 10% carbon dioxide. Mixture 2 was with 60% methane and 40% carbon dioxide, and mixture 3 contains 5% methane and 5% carbon dioxide and 6% oxygen. The nitrogen gas was used as the balanced gas in the above mixture for calibration. Here is the generalized flow diagram of anaerobic decomposition of organic matter and generation of methane. Firstly, complex polymers, which include cellulose, polysaccharides, and proteins, they undergo hydrolytic fermentation bacteria and monomers of fatty acids, sugar, amino acids, NH3, hydrogen sulfide, carbon dioxide, acetate, hydrogen, like these are produced. Then comes syntrophic entogenic bacteria, and these are all are converted into acetates, hydrogen, and carbon dioxide. Then methanogenic bacteria works and acetate, hydrogen, and carbon dioxide converted into methane plus carbon dioxide. Properties of coal washery rejects. Coal washery rejects were obtained by washing the subbituminous coal. The ultimate analysis of the coal washery rejects revealed that it has low carbon 21.9% and oxygen content 7.2% compared to that present in raw coal obtained from the coal mines. This less carbon content makes it inappropriate for the combustion process. Thus, it is being discharged as waste. but the recovery of the remaining carbon present in it is possible in the biogenic method. Then comes the method 2, which is improved formation of biogenic methane by cultivable bacteria in highly volatile bituminous coals. Coal methane CBM is an important energy source for the global energy transition. The utilization of CBM has a much lower impact on humans and the environment than those of the coal and oil. Side description of coal samples. Coal samples were collected from the 1412A mining working phase of the coal mine. The coal samples were taken 10 to 20 meters away from the coal wall as cylinders with a diameter of 108 millimeter and length of 80 to 100 millimeter. Sorry. The microscopic coal petrology of the samples was that of dark bright coal, vitrinite 63.5%, inertinite 25.40%, Exenite 11.10% and total carbon 92.79%. The coal quality was highly volatile bituminous coal 1.93%, air dried, basis moisture 10.77% air dried. Then comes enhanced gas production by the inoculation of cultivable bacteria. A total of 3 coal microorganism groups were inoculated for 50 days. There was no significant methane or hydrogen production in the water group, and the methane concentration of the water group peaked at 0.16 plus minus 0.01% on the 14th day. The concentration dynamics of the methane and hydrogen were indeed very similar between the treat group and the medium group, both showing an X shape. Initially, hydrogen concentration was increased rapidly, methane began to increase. Then comes the improvement of the biogenic gas. Biogenic gas production can be improved by the activating coal microorganisms, which is of great significance and application value to increase the production of the coal bed methane. However, it remains poorly understood about the biological mechanism of the cultivable bacteria in coals that enhance the gas production. Cultivable bacteria in coal were enriched by minimum salts plus yeast medium, and coal was cultured with these medium removed bacteria cells in minimal cells medium. Metagenomics were used to analyze the differences in the microbial composition and function. The results showed that cultivable bacteria caused large changes in the microbial and the functional structure in the coal, and the cultivable bacteria Bacillus dominated in the treated coal. Metagenomics analysis showed that the K is easy orthologous from 4-methanogenesis pathways in cultivable bacteria treated coals were detected. These pathways were carbon dioxide to methane, methanol to methane, acetate to methane, and using methyl as the terminal methyl carrier. Importantly, the cultivable bacteria mainly increased the functional genes related to the degradation of the benzoate, fatty acids, fluorobenzoate, xylene, and aromatic compound, and further increased the acetate content. In addition, metagenome assembled genome analysis showed that the PTA and CISK genes mainly originated from the bacillus, which was then beneficial to acetate production. And the cultivable bacteria treated coals had a higher methane emission than the control groups. In summary, cultivable bacteria can enhance the biogas production by enhancing the organic matter degradation and acetate pathways, which is of greater importance for increasing coal bed methane production by domestication. Then comes the conclusion. The relationship of the microbial species with the coal environment are of the critical factors for achieving the increased coal degradation and utilization. As they appear to be relatively favorable microbial substrates, the heterogeneous and the aromatically condensed structure of the coal cannot be regarded as completely understood in terms of the bioavailability and bioefficiency due to the complexity of the coal microbial interactions. The subsequent application of various microorganisms to benefit coal largely depends on their metabolic and functional characteristics. Therefore, establishing highly adaptive and selective microbial groups may help to determine rate limiting steps and enhance the prospects of the bioutilization. Furthermore, despite the lengthy research history and promising reports regarding the efficiency of the indigenous microorganisms, there is still no precise prescription for successful technology innovation in a practical large scale application. What are the future trends? First, until now, fundamental research on coal biodegradation and bioutilization has focused mainly on the laboratory scale screening of the various methodologies and exogenous microorganisms. However, the implementation and optimization of these processes using indigenous microorganisms in full scale outdoor systems remains attractive. Many outcomes are too preliminary to predict the details of a commercialized process. Some consideration must be given to upgrading the technology to bring these processes to regulatory issues and policy that may exert a strong influence in the future. Because coal bioutilization is a complex and intricate process, efficient organisms and processes will be critical for economic competitiveness. The modern techniques of recombinant DNA technology and protein science may serve as enhanced tools for the manipulation of indigenous microorganisms. Furthermore, exogenous coal solubilizing bacteria may become part of the indigenous microbial consortia that colonize coal environments, though they may be able to naturally thrive and subsequently solubilize coal in this ecological niche. And then comes the reference of my article. Thank you. That's all from my side.